Hey there, and welcome back. For centuries, we've known that we get traits from our parents, right? But what was actually passing that information along? What molecule held the blueprint for life? For the longest time, it was a total mystery. Well, today, we're going to solve that biological whodunit. You see, back in the middle of the 20th century, this was the biggest question in all of biology. Scientists knew there was some kind of genetic material, a master plan tucked inside ourselves, but they had no clue what it was actually made of. So let's dive into the incredible detective story of how they finally cracked the code. Okay, so there were two main suspects on the table. In one corner, you had proteins, and honestly, they seemed like the obvious choice. They're incredibly complex, built from 20 different building blocks. It just made sense that something so complicated could carry complex instructions. And then there was DNA. It seemed, well, a little boring. Kind of simple, with just 4 repeating parts. Most scientists were putting all their money on protein. And that's where our detectives come in, Alfred Hershey and Martha Chase. The year is 1952, and they are about to design one of the most brilliant and elegant experiments in the history of science to settle this debate once and for all. They called it the blender experiment. And the goal? It was pretty clever, actually. They needed a way to follow both of our suspects, protein and DNA, to see which one was really calling the shots during a cellular takeover. Their key tool was this tiny virus called a bacteriophage. Now, you can think of it like a little microscopic syringe. It latches onto a bacterium, and it injects only its genetic material, leaving its outer shell behind. That material then hijacks the poor bacterium and turns it into a virus-making factory. And Hershey and Chase had this aha moment. If they could just figure out what the phage was injecting, they'd have their culprit. But wait a minute. How in the world do you track molecules that are completely invisible? Simple. You make them glow with radioactivity. The plan was to cook up 2 separate batches of these viruses. In one batch, they'd tag the proteins, and in the other, they'd tag the DNA. And this is where the real genius of the experiment kicks in. It just so happens that proteins contain the element sulfur, but DNA doesn't. And on the flip side, DNA has a phosphorus backbone, which proteins totally lack. This was the perfect chemical loophole. It allowed them to use radioactive sulfur to label only the protein coat and radioactive phosphorus to label only the DNA. They now had 2 perfect, unique trackers. So the whole experiment itself boiled down to this really beautiful 3-step process. First, you let the viruses do their thing and infect the bacteria. Second, you pop them in a blender to knock the virus shells off the outside. And third, you use a centrifuge to split the heavy bacteria from the lighter virus bits. All right, let's break it down. Step 1 is infection. Over on the left, you've got the viruses, where the protein coat is tagged with that radioactive sulfur. And on the right, it's the DNA inside that's got the radioactive phosphorus tag. In both scenarios, the viruses latch on and inject, well, whatever their genetic stuff is. Next up is the step that actually gave the experiment its famous name. They literally put the whole mixture into a blender. Now, this wasn't to make a bacterial smoothie, obviously. The agitation was just enough to shake those empty virus shells, the little protein capsules, right off the surface of the bacteria without bursting the cells open. And for the grand finale, centrifugation. They spin these test tubes like crazy. And since the bacteria are heavier, they get packed down into this dense little clump at the bottom. That's called the pellet. The much lighter, empty virus parts are left floating around in the liquid up top, which we call the supernatant. All that was left to do was to see where the glow ended up. So, this is it. The moment of truth. Where did the radioactivity end up? Was it in the pellet with the bacteria, which would mean the molecule got inside? Or was it in the supernatant with all the empty virus shells? The location of that glow would finally reveal the identity of the genetic material. First up, the verdict on our main suspect, protein. In that experiment, the one with the radioactive sulfur, almost all of the radioactivity was found floating in the supernatant. What does that mean? It means the protein coat never even made it inside the bacteria. It was just an empty jacket left at the door. So suspect number 1 was officially cleared. But then, this is the discovery. The eureka moment. In the second experiment, the one with the radio-tagged phosphorus, the glow was right there in the pellet, smack dab inside the bacteria at the bottom of the tube. This was the smoking gun. The DNA had successfully entered the cells. That was the stuff that started the takeover. The conclusion was just undeniable. It was absolute, unequivocal proof. DNA,
The molecule everyone thought was too simple for the job was, in fact, the one. It was the master blueprint of life.
So, case closed, right? Well, yes and no. This amazing discovery just blew the doors open to a whole new world of questions. Like, what is it about DNA that makes it so darn good at its job? And what about its molecular cousin, RNA? How does it stack up?
You know, it turns out that being the genetic material is a pretty demanding gig. To even qualify for the job, a molecule has to check a few boxes. It's got to be able to copy itself perfectly, be stable enough to last for generations, allow for little changes, mutations for evolution to work with, and finally, it has to actually express the traits it's carrying.
So when you put DNA and RNA head-to-head, the differences are really striking. Look at that stability. DNA's double helix structure makes it rock solid, perfect for storing information safely for a long, long time. RNA, on the other hand, is less stable, it's a bit more reactive, and because of that, it also tends to mutate faster. It all boils down to their primary roles. DNA is the vault, the secure archive for our genetic code, while RNA is more like a quick memo, a temporary messenger.
Yeah, the best way to think about it is like this. DNA is the master blueprint, right? It's locked away for safekeeping in the cell's nucleus. RNA is like the photocopy you take to the construction site. It's the working copy that gets sent out to the cellular machinery to get things done. So DNA is built for storage, RNA is built for action and transmission.
But this all brings up a really mind-bending question about the origins of life. I mean, if DNA is the superior storage molecule now, does that mean it was always in charge? Or did life maybe start with something a little different?
And this leads us to this really cool and compelling idea known as the RNA world hypothesis. The theory goes that way, way back before the whole DNA-based system we have today evolved, the very first life on Earth used RNA for everything. And why do scientists think this? Well, it's because RNA is kind of like a molecular Swiss Army knife. It can store genetic information, just like DNA, but, and this is the key part, it can also fold up into complex shapes and act as a catalyst to drive chemical reactions, a job that proteins mostly handle today. Because it could do both of these essential jobs, it's the perfect candidate for a single molecule that could have gotten the whole show of life started.
So the Hershey-Chase experiment basically handed us the script of life. It proved it was written in the language of DNA, but finding that script, as absolutely huge as it was really just the beginning. It was the key that unlocked the door to understanding how those genetic instructions are read, interpreted, and ultimately transformed into the magnificent, mind-boggling diversity of life we see all around us. The story, as they say, was far from over.